Good evening, everyone. So, from today onwards, we are going to start a new module that is R programming. Okay, so in next few days, we will deep dive into this R, which is very powerful language and which is used for bioinformatic application, even data analysis, statistical computing. So, let's understand how we are going to utilize this for our application. Okay. So, first, let me know how many of you aware about this prior, like worked in R programming or any package already used for your analysis, or you all are new for this? Anyone? Just text in chat box if you are aware about R programming. Otherwise, we need to start from scratch. Uh, yes. Any idea about R programming? So we are analyzing this genomic data. Okay, no idea. Fine. Or even we can use for a visualization purpose. Like R is itself a wide range of tools, libraries, packages, we can say. So which are very useful for bioinformatic workflows and bioinformatic data analysis. Okay. So at the end of this module, when we'll complete this by at least 8 to 10 sessions we need for this all things. So you'll be comfortable in handling data, you're comfortable in running analysis, even generate visual like visualization, like generating graphs, plots, and every detail you'll get to know. And even if you are not getting any point at any level, please free to ask. Okay. So, okay. So let's start. First, why we need any kind of such programming language? Like what exactly the use of programming language? We already learned this last module, that is Python, right? So basically, if you consider like functionality of R, so it is divided into three different classes. Like if we summarize what exactly the role with respect to this R language, like as a bioinformatician, as a statistician, or a data science, any role you consider. So in general, first step always is data interaction. We need to interact with the data. Whatever data we are getting, we need to analyze that data and then a result visualization or interpretation. Okay, so in data science or in context of bioinformatics, so this is nothing but like art, how you're extracting knowledge, how you understand the biological data. Okay, so this is or this is a basically introductory session, the first, like how to explore this R, how to understand the application R in data science, introduction in bioinformatics, some basics for R language, and application in genomics. So these all things we are going to cover, and then in hands-on session we will cover from scratch, from installation, the different types of platform, environment, and basic syntax commands, data handling, manipulation, how to import export data, visualization, biological sequence analysis, like how to work with FASTA sequence, how to import that sequence, and many more things we are going to cover. Okay, so first let's understand R, uh, like in data science for bioinformatics both way. uh, Anyone have any question? Okay, so yeah. So R itself is a very versatile language, open source language. So we'll go through the first overview, its application, what all different tools, functions, which are specifically tailored for biological data, analysis of biological data. Now, the most important question, like why data analysis is so much crucial with respect to genomic data. You may ask this question. Yeah. So genomic data means what exactly you mean. Genomic data, like that data might be the sequence data, any complex structures. So first thing, through this data analysis technique, we can decode the pattern, we can identify the trends, even we can understand or draw some meaningful conclusion. Right? So it is advancing understanding of this genetics, and also we contribute to find some discoveries. So first understand R in bioinformatics. So R, as I told, it is very powerful language which is widely used in bioinformatics for statistical analysis, like whatever numerical data we have, we can do statistical, we can apply formulas, even we can visualize the data. So it's a versatile environment where we can handle biological data, complex biological data. Okay, then analysis capabilities. So analysis capabilities, because our mm, our programming language excels in data analysis, also offers like functions like how to process the data, how to interpret large data set, and it's make essential part in genomics, and also not only genomics, but yeah, you can consider in any omics, genomics, proteomics, any omics study. Bioinformatic packages in R. So packages, specialized packages like you can say a bioconductor, which is specifically made for handling biological data, also provides tools for genomic data analysis, sequence alignment, and many bioinformatic tasks. Visualization. So visualization, again, there is a base function, plot and all this, but there are specific libraries called as a ggplot, which enable to create detailed, or we can say informative graphs, like paper or publication quality graphs, which helps to present our biological data more clearly, more effectively. Okay, so we can interpret the result very clearly. And also integration with bioinformatic tools. So integration means like bioinformatic tools and database, we can interact in the same environment where we are working, and also we can streamline workflows, even we can efficiently process the data, analyze the data. So let's understand basic, as we need to write code which is understandable by that environment, our environment. So we must know some syntax and structure, how we can write code, as R itself has clear code and readable syntax. It's not much difficult as compared to other programming languages like advanced languages. So here you can see syntax and structure, what exactly this. So syntax means where we suppose we want to assign some sequence with any variable, or we want to generate function or apply function, same code we want to execute, even control structure is there. So understand the structure is most important part, and which is very essential for handling or analysis of the data. Then data structures. So R supports various data structure, like there's a vector, there's a list, matrix, data frame. We'll see in detail, we'll do hands-on session with each of the structure, how to handle, how to store data, what kind of data we can store in that structure. So everything we are going to perform. So each structure basically serves specific purpose. Like you have a sequence data. So sequence is nothing but all character, ATGC or amino acid sequence, any sequence you can consider. So these are like text data or character data. So what kind of data structure you can use for storing that kind of data. Even if you're handling any numerical data, gene expression values, or count, gene count. So that numerical data we can store. In some cases, you need to store both kind of data, like character data, also numerical data. So what kind of data structure you need you basically prefer. So these all things we will learn. So data structure basically, it serves the purpose, like how to organize your data, how to manipulate your data. Okay, like your wardrobe, you can consider where you can store every element, every atom separately, properly. So that is nothing but structuring or analyzing, organizing your wardrobe. So the same way, if you have a huge amount of data, how to store that data in a proper way, so whenever you need that data, you can fetch efficiently. Again, there's a term like functions in R. So functions is like, it's like automated thing where you can use same code for multiple elements, or basically it's, you can use this for small task like simple calculation or any complex analysis you can perform. Again, in function there are two types: built-in function and custom or user defined functions. So built-in functions are already there in environment, they are just like a pre-built tool we can directly use, we can call that functions. And user defined, that is like customized as per the experimental requirement, as per the user need. Okay, so we'll see how to create functions, user defined functions. Then there's a libraries in R. So libraries, like again, it's like a package. So there are different packages specifically, I mean, for biological data analysis, like or visualization purpose, like ggplot is there. For manipulation, there is a dplyr, data manipulation, then tidyverse, hr, dsec for no, downstream analysis, normalization, and all these things. So they are specific, we can say specific functions and specific tools for data manipulation or any particular task. We can use that particular package, and in that package there are multiple functions, so it's like a compact package of related functions. Then we'll see your application in bioinformatics and in genomics. Specific application, as specific in the sense, basically R plays a very crucial role in genomic research, like from pre-processing of your data. Nowadays we are getting raw data from sequencer machines, that raw sequences. Okay, so that raw sequences are very huge in quantity, are bulk data. So first we need to pre-process that data, because on huge data set we cannot perform any like we need to first filter that data. Suppose some ambiguity, some missing values are there, we need to clear all this stuff, right? So pre-processing is very important step, and for this all kind of data we can use this step for cleaning of purpose. Then if anybody looking for variation, genetic variation or expression pattern, then these are specific application. Visualization, so visualization we can create meaningful or very good quality graphs, charts, then plots. Okay, so when we want to interpret any complex genomic data, genomic information, we can visualize the data, it is very clear to interpret the result. Analysis, so analysis in the genomics basically. So suppose we are looking for differential expression analysis, we are looking for variant calling, or just want to identify correlation with some large scale data set, genomic data set. So statistical analysis or advanced machine learning part is there. Machine learning in genomics, nowadays it is very useful in market, AI, ML. So when we want to use this part, we should know some algorithms and uh, the logic behind that algorithms. Suppose we want to use predictive modeling, classification, clustering, even a pattern recognition. So these all things we can use in genomics. And for example, we can use in personalized medicine or biomarker discovery, these all techniques. Then along with advantages, there are some challenges as well. So let's see. So some unique challenges, like due to some complexity or scale of dataset, because this data set is generated by advanced sequencing technologies. So here you can see the dealing with large scale genomic data set. So large scale data set, large volume of this all data, genomic data, like whole genome sequencing or any transcriptomics. So this is a present challenge, how to store this data, after storing how to process that data, and then analysis part will be there. So storing is important. If you store properly, organized way, so it is very easy to fetch that data. If you just fire a query, so to fetch that particular element, particular data, so it is easily you can fetch if it is organized properly. So storing, then processing, and then last will be the analysis part. Complexity and heterogeneity, basically complexity and heterogeneity means there is a diverse or different types of data, various types of data, like in sequencing itself, suppose you are working with the DNA sequence or RNA sequence, epigenetics. So these are different kinds of data which require different analysis approach. So this is again a challenge to handle such kind of heterogeneous data. Computational and storage demand. So to process this all genomic data, we need resources. We definitely require efficient algorithms so we can handle that data properly, and also infrastructure, proper structure, data structure we need so we can store this data and then we can handle the data properly. Efficient tools. So as itself are is powerful statistical programming language, but still like it should have some packages, advanced packages to handle manipulation, analysis, visualization, these all things. Parallel computing and optimization, like like uh, if you want to enhance scalability of this genomic data analysis, then parallel computing is must. And reproducibility and documentation. So when we work with this genomic analysis with R, so we need to maintain reproducibility, right? So R, first thing, the code transparency is there, then documentation support will be there. So these all things matters. So let's see how to install this in your system. Okay, so I'll share the link. You just click on that link and I'll guide you how to download. Till here, any query?
No, ma'am.
Okay, so just let me share link. Okay, I pasted the link in chat box. Kindly open this.
Ma'am, you are not audible.
Oh, sorry. So I was telling, just here you need to click on this download R. Here it will navigate to the page where you need to select the operating system, and then it will navigate to this page. So you just click on this download. This is the latest version. It will download exe file, and you just need to click as you installed any app same way. Okay, so what exactly this are, this what we have installing right now,
Ma'am,
that is
wait a
our.
minute. Can you just explain again, please,
Yeah,
please,
yeah, yeah, sure.
slowly, because actually we can't hear previously.
Okay, so you need to click on this option download. Here you'll get this option download and install R. Okay, so you can see here Windows and Mac user likely want one of this version. So here these are the three options are given for Linux. Otherwise you can go with, or if you have a Windows system, you can just click on this download R for Windows. It will redirect to this page subdirectories, and you just need to click on base. So in base you'll get this option download this link from where you can download the file .exe file. So our installation is very easy. Like, it's like depending on your operating system, you can select that link and just go through the steps. So when you download this, in next step, till here done.
Yes, ma'am.
Okay, so you just need to select path where you want to download, and by default it selects C drive, because most of the softwares we need to save in our C drive. So don't change the default setting. Just select whatever provided. Just select next. Select all the components which you want to install, and then just proceed to the next. They are auto selected, so no need to select manually.
Ma'am, there is two option in the startup option.
Hmm.
Yes or no. Yes for the customized startup and the no for the accept default. Mm.
Except no need to customize anything.
Okay, ma'am.
Just accept
As
all.
it is, I'm just downloading what the option is
Yes, yes,
already selected. Okay.
yes. Accept default, and when you proceed to the next, like installation, it will start that progress extracting file and installation step. And at last you just need to click on finish, so it successfully installed R in your system. So how to check whether it is installed or not installed? Okay. So first thing, when you're done with the installation step, all of you done?
Yes.
Okay. So you can just check on your desktop. On your desktop, there will be icon that the version R 4.4.2. You just need to click on that icon, so it will open like this. R base. Just let me know it's working.
Yes, ma'am.
Okay. So this is called as a R base, R software. Okay. So what exactly this and how to use this, how to navigate this? So here, let's understand first. This is called as a script area. Like, here you need to write, here you need to like, this is console itself, but whatever script you are going to write in this, like, let me give one simple example. 2 plus 3, if we are doing this at uh, calculation, and when I click enter, it will give me output like 5. Even if you want to execute any statement by using print, so we need to type the command, type the function, every detail. Like, there is no auto detection, no auto completion. Okay, you have to type this. Like, suppose we are writing or just executing high, so in print function we can execute this. Okay, then if like, here you can see file, you can explore this, source, new script, open script, display different files and history, these all things, exit. Even in edit, there are different options: copy paste, visualize toolbar, status bar, packages. So there is a package cran and install packages. So if I want to install something in this environment, in this software R base, so I need to type command like this: install, or the command is install dot package. If I want to install single package, then I need to write package. If there are multiple packages, then packages. In bracket we need to mention name of the package, and this is the way. There is no shortcut, there is no graphical user interface where you can click and install that particular thing. Okay, even if you want to visualize something, like you want to generate plot on any data, so you need to fetch that data set by using plot function, and then whatever plot are generated, you can download that and then reuse. Then these are different options: style, visualization, and help, save. So this is only one space where you can write, you can visualize. Here you write code, and output is also in the same console, in the same space. Okay, so now time to download the second option, that is RStudio. The same step you just follow. Click on this download RStudio desktop for Windows and complete the process till finish. And I'll wait for till that time. Just proceed this.
Can I know what is the difference between R and R
Yeah,
Studio?
yeah, yeah. I will tell definitely. Uh, just complete the steps. So practically, we'll see what exactly the difference and which one is means user friendly. Yeah. So basically, our base is a software. This one, first one. Our studio is a IDE. IDE is integrated development environment. Okay, so this is specifically designed for R. Means it is more user friendly, like because of its interface, having different tools, having different features. Like if you are new to this, then definitely it is easy to navigate in R Studio. So let's first install this R Studio and then we'll explore. First, you need to verify that you already installed R. So this is the first step, and then you can use RStudio. If you not, if you don't have this R base, then you cannot use this RStudio. Okay, so first, because this is a software, software is must to run IDE. Here also you can work only in R base, but you have to type each and everything manually. Okay, let me open R base. So whatever fundamental functionalities are required, or core features for our language, these all are available in this R base. This is called as a, let me write it here. If I want to write something which is not for execution, then I can use comment. So comment is the part just for our understanding, not for execution. Suppose this is what CLI. CLI means command line interface. This is interface where we have to write each and every command. Okay, so this is the, like here we wrote this 2 plus 3, we have executed this print high, then it will execute. So we need to write this all thing command, we have to provide command here. And this is a R console, you can see here R console. So scripting and everything we can perform in this R console, whatever writing execution part or any sequences, continuous code we want to execute one by one. So these all things we have to write and plain text as a series of R commands, and we can execute in this R console itself. Done with R Studio.
Yes, ma'am.
So if you complete the down, download step. So that is a app. Our studio is a app. Our base is a software. Okay. So when you want to search for any app, you can just explore here in search bar. Just type R Studio. Here you'll get app R Studio. Okay, just click on this app and it will open like this. Okay, first open this and then I'll explain how to work in this. So this is very user friendly interface. Like this is called as a IDE, integrated development environment. Like it's having simply we can say it's having a point and click interface. Why? Because we can just point whatever we want and click, it will execute. No need to type. Yeah, we need to type code, but it's not like every, like suppose let me done, installation part is done.
Yes.
Okay, so we can explore here. If we want to write, edit, execute, is all thing we can do here in first pane. These are called as a pane, P-A-N-E, pane, or let me show here, view panes. So this is by default structure. Like here you can see the left top area is for script editor, script writing. Then this right hand side is the environment, history, connection, tutorial. Then bottom left is for console. Then the bottom right is for fetching different files, plot, packages, and all this thing. But yeah, you can change the pane. Like go to the second here in view, go to panes. You can switch in console on left and console on right. So let me show this. This is on right. So what is the difference and which one is comfortable? It's up to it's like a personal choice where you want to keep. I'm keeping console on my right hand side because if I execute any code, this is a this is script editor where you can write script like the same script. If I write here 2 plus 3, okay, so this is editor where you can write, you can edit, but for execution you can just click on this option, run the current line, which is selected. Selected means where cursor is blinking, or you can just select properly like this. Here you'll get numbers as well for each line. Now I'm just executing this first line. So the output we are getting here in console region. Okay. So console is what? This in RStudio basically it is integrated console where for direct interaction we can say like here you can see terminal. We can interact with our system terminal directly. Even background jobs, if you are working with multiple projects, so simultaneously you can run different jobs at the background, and currently you can work on your current status, whatever work. Okay, so this is a console. Now let me show in the script editor where if I press enter, you can see the second number line. Suppose I want to write the same command, that is print command. If I use here PRI, you'll get different options available. This is auto completion, code completion. So yeah, we need to select this print. We want to execute. So I'm selecting print. Okay, so this is a function, and function always comes with parenthesis. So here if we need to write something, if you want to execute any line, you can write. Otherwise, if you want to execute any variable or value, like here I'm executing high, and if I there's one shortcut, you can use control enter key. Here you'll get this idea. Control plus enter. You can read this. Run the current line by using control plus enter. So this is easy. So you'll get output in console. Okay, even if you consider this 2 plus 3 with you want to save this with some variable like x is equal to 2 plus 3. So first I need to execute this line, then I can execute by using print function. Like if I want to execute this x, here is x. Okay, so X is new object generated. So in environment, it is generated. The value is X. That object is generated with assigned value. Here you'll get all details. This object is numeric, or you can say variable. Okay, variable which hold the data, that is numeric and holding 56 bytes. This is the actual value. Okay, if you don't want this in objects in your current workspace, or any previous objects are there, so you can clear these all objects by just clicking on this clear option. You can remove all objects. This is third pane, environment. Even you can import the data set. You can check history which previously executed code. You can check here from here. Even connections, you can connect database. If you want to learn something through tutorial, you can learn. This is for basically learning purpose. Okay, now what is the difference between then the script editor? Whatever we are doing here, the same thing we can do here, like this. 2 plus 3. Let's execute in console, as we already executed this command in our base, right? So if I execute here by just clicking enter, we are getting answer. Then why there are two different panes like script editor and console? What you think? The same task we can perform, right? Okay, so in our base, the same thing, console is there, right? So in our base, script execution, we did through plain text file. Okay, there is no advanced code editing features. But in R Studio, here you'll get script editor, like syntax highlighting is available, code completion is available. Even you can check errors. You can enhance this all experience. Okay, so here in our studio there is a separate console, and if I erase everything, like from here we can erase, this is the option, clear console, or you can use shortcut control plus L. If I erase, it is not like regain, it's like lost. But in our script here, you'll get an option, you can save and reuse the same script for different purpose. Suppose I just want to save this with demo name. So save as type R means it will save by R extension. Like here you can see demo.r, and if I open this, you'll get this file. If I double click on this, it will open in a new window. Or if you want to open any previous file which you have created, that also you can open in this script editor. You can create multiple files, like from just clicking this plus sign, or just go to file, new R script by using control shift N. Otherwise simply you can just click R script. Multiple files you can open, you can work simultaneously with multiple scripts. Okay, and you can save as well. Like suppose I want to save this now with demo 1. So this is another file. If I close from here, but I want to open, so I can open like this, it will open. So you can work multiple files simultaneously. Now this part is clear. If you are working here, your cursor will blink. If you just want to switch directly to the console, you can use shortcut control 1 and 2. So if you use 2, it will switch to console. If you control 1, if you can switch to script editor part. Now let's explore this fourth pane, which is important. The first is for files. You can see all the files from current working directory. So how to set your working directory? So for that, you need to go here in session, set working directory, which is your current working directory. Means suppose you have some files, Excel files or FASTA files, you want to work with that files. So directly you can set that folder as your current working directory, so you can fetch files in your current working environment script. So select choose directory, From here you can select which one is your current working directory. So I've selected this. Suppose you want to select Python to explore Python files, the last module which we have covered. Okay, so like this you can set current, or there is one more option. If you want to check in which directory you are currently working, so check with get working directory, so it will show current working directory. If you want to change this, then let's change by using set working directory function. So set working directory means you have to set path. So to set path, you can, I'm just taking help of this path. In double quotes, you just mention the path and then write the path. So I think there is a R, like this is this directory I want to set as my current working directory. So now execute line number 1 to know current working directory. So now it is R, okay, which having these two files. So this is for files. You can navigate plots. Here, if you generate any plot, like by using plot function, so this plot is generated in this fourth pane. You can export these plots, like either you can save as an image, you can download as a PDF file, or you can copy to the clipboard. For packages, you can switch to the third tab. Here you'll get different package list. From these all package list, you can search if you're looking for visualization plot package, ggplot. You can see here this package is available. This is the description also you'll get, like this is a grammar of graphics where you can generate elegant data visualization or different plots. And you can directly install by clicking on this install button. No need to write command and all those things. So it will install from repository. Here you need to type package name. You can click install dependencies. We will see in detail this part how to work with packages. Okay, even if you want to use command line interface, like I want to write command, so here you'll get code completion install dot packages or packages, these two options. Install packages if you want to check, select and just mention the name of the package. Like this you can install. If you don't want to execute this line, so just comment this. So this is just for our understanding, not for execution. Okay, then there is one more tab, help. So help is basically to suppose if you're not aware about any function, if you're not aware, like here we already used print function, right? But you don't know what exactly usage of this print. So you can take help by just writing question mark and type the name of any function or any command, execute. So here you'll get description. Print is a base function. Description is what? Print, its argument and return. So what all things we can use, how to write syntax. X is the argument along with other argument. Like these are optional arguments you can mention. And there are many arguments are given along with description. Also usage, references, every detail you'll get in help. Okay, you can also search for get working directory, how to use that. So like these all things, this is just okay, get or set working directory. So usage and all detailed description you'll get here. Then there's a viewer and presentation. So viewer is just to check or visualize the history, okay, what you can say, presentation, whatever we did, that we can visualize here. And also we can create our own presentation like PPT presentation in this part. Okay, what else? Yeah, so here these all different tabs we will explore in hands-on session. So the basic is clear. How, what is the difference between R base, R studio? Okay, so our base basically, this is a community support is available. Okay, but may lack the resources, may lack the integrated environment. And here in our studio also you'll get a strong community, like different additional resources are available, active support system is available. So in short, we can differentiate between our base and our studio. Is like our base is a software which contains our programming language, where our studio is software that makes our programming easier, and both are totally free, open source. Clear? We have not used this viewer, but basically this is for interactive or render documents or any web-based content. If you want to visualize, we can go with this tab. So the purpose of script editor is for primary workspace, where you can write, where you can edit. Also it comes with feature syntax highlighting, code completion, error checking, or you'll get these line numbers. And code folding is also there. So code folding you will learn in control structure how exactly it works. Even you can use this function like find and replace. If you want to search something or any term, any you want to replace suppose any particular word, so find and replace you can use here in script editor. In console, this is again interactive interface where you can directly enter and execute our commands. Okay, you can check your in history, but from here if you delete, you cannot replace the code. Okay, so environment is basically for history and also connection purpose. We can use this, whatever the objects we have generated, we can check that objects. Even we can import the data set, import data set, like if you want to view any existing data set or work with any data set, so we can import that from system. Okay, so these are all things about our base and our studio. And this is for comment. You can use one line comment by using hash. This is one line comment. We can also create multiple lines comment. Suppose we want to use here any multiple lines, so we can use this. Okay, we will explore this in next session. Any doubt till here?
I'd like to ask a question.
Yeah, sure.
Actually, I haven't. This is my first time using R, and I'm not aware of the codings and everything. Do you have
Okay.
any material that I could refer to understand codings and functions?
Yeah, share. I'll share these all things. When we'll start actual coding, I'll give you script. I'll give you if you want any document, we'll share. Okay. Just be
Mhm.
comfortable with interface. As today is our the first session, like how to navigate through this interface. So be comfortable with this interface with R base and R studio. You can try different like mathematical formulas like 2 plus 3 as we already use such way. You can use print statement to execute and these all things with R base and R studio. And
Another
from tomorrow onwards, we'll start from scratch, like how to start writing code from scratch. Okay, if you are new in programming, then don't worry about this. We will make it comfortable, everyone. Yeah. Any other query?
also, actually, in my